Starting to record. Okay, can everybody see where it says sports? Anybody who can't unmute your mic and say something. I can't really see what you guys are doing when I've got this on my monitor.
It's good.
It's good?
Mm-hmm.
Okay. Awesome cakes. All right. Yeah, I thought we should talk a little bit about sports. There's not an awful lot of live sports to watch, but at least I've got some nice pictures. But, just as I use beer as my sneaky way of introducing enzymes, I want to use sports to talk about mutations. This is another one of the things you can see from the top of the mountain that we've climbed. We've talked about DNA replication and how it's replicated by a set of enzymes called DNA polymerases. We don't really need to know for our purposes how they work. You can just think of them as little, knitting machines that put nucleotides together and weld new strands of DNA. There's a whole nother set of enzymes that repairs DNA damage. DNA will break if you look at it cross-eyed. It's a pretty fragile molecule. And so you've got a bunch of DNA enzymes that are constantly going down your DNA strands, looking for damage, snipping out damaged parts and rebuilding them, and fixing breaks and things like that. And those enzymes are really good, but they're not perfect. Every so often they'll make a mistake. And the mistake can be as simple as, one T gets replaced with a C, or an A with a G, or a T with an A, or what have you. Or the gene, sometimes you can have single nucleotides or 2 or 3 cut out. You can even have a gene that ends up silenced. It may no longer be transcribed or translated at all. One moment, somebody's joined us. There. Okay, Francisco Martinez has joined us. You with us? Okay, I am, all right. I'm seeing Francisco Martinez. I'm going to assume he can see us. Okay, when we take breaks, I'm going to check the chat in case I can't hear your voice or you can't hear mine, but you can always go to the chat log. Okay, where we were. All right, can everybody see this again? Somebody say something?
Yes, it's good.
Okay, great.
Thank you.
Okay. You can also have errors in that crossing over process that we talked about during meiosis, and you can have portions of genes or even whole sequences of genes getting duplicated, getting deleted, getting spliced in weird ways. And all of these are considered mutations. Mutation is just any heritable change in a DNA sequence. And anything that can cause mutations is called a mutagen. There are chemicals that will do it. Radiation will do it. Ultraviolet light will do it. X-rays will do it. All of those things can damage DNA. And the repair enzymes do a very good job, but they're not always perfect. And sometimes they will leave a mistake in. They won't catch something. What you're looking at on the left is a dish of nutrient material used for growing bacteria. And every one of those white spots is a colony of millions of single bacteria. We use dishes like this in what's called the Ames test. You basically expose bacteria to something that might be a mutagen, grow them in culture, and see how many mutations that you get. We don't really need to know the details of how it works, but it's the first line test for mutagenic activity in new chemicals and new consumer products. If you're going to sell a new line of, I don't know, lipstick, and you are concerned that it might cause cancer, the first test you would do would be to expose some bacteria to lipstick and then run the Ames test looking for mutations. You'd still probably have to test it in, other creatures, maybe on rabbits or something like that. But the Ames test is much cheaper, much less morally compromising, and much quicker. So the Ames test is the standard first line of testing for mutagenic activity. The reason why this matters is cells control their division with proteins that you can think of as being brakes and accelerators. Exactly how they work, we don't need to get into, and it's not really my field. We've got other people in our department to ask if you're curious. But you can think of these proteins as being, switching on the division process or blocking it from happening. You also have a fact that cells that are damaged or cells that aren't needed undergo this very orderly process of putting the chairs up on the tables, mopping the floor, shutting out the lights, metaphorically. They don't literally do that, but they shut down and they break up. And we call this process apoptosis, otherwise known as programmed cell death. If you want an example of it, when you were about the size of a bean in your mother's uterus, your hands look like paddles, and the cells that used to be here, 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 and here between your fingers got a signal that made them commit this orderly form of suicide. If they didn't do that, you would have webbed fingers, but assuming that they did die as appropriate, that's why you have separate fingers. The cells that were in between underwent apoptosis. And this is also controlled by certain genes. What I'm talking about is an entire course in itself, and even then we don't understand everything about it. This is a very hot field. Let's just say that some genes code for proteins. Well, genes code for proteins, but in some cases the protein can go back to the DNA and can either block another gene from being transcribed, or it can encourage a gene to be transcribed. That's a picture of what it looks like. In blue and green, you see the DNA, and blue and red is DNA. That green thing with lots of alpha helices is a protein that binds right to the DNA, and this particular one will block other genes from being transcribed. The upshot is that genes can work almost like circuitry, where many genes produce proteins that go back and block or promote other genes. So genes can act as on-off switches. And this is a small part of how genes control things like division and apoptosis. If the control genes, the ones that are controlling how often the cell divides, undergo a mutation, they may not function the way they should. If the genes that cause a cell to undergo apoptosis, the kind of kill switch genes, if they can no longer function as they should, the cell may not be able to die when it's time. And if a cell is dividing uncontrollably and can't stop itself and no longer has that kill switch, we call it cancer. And those damaged genes that result in cell division that's out of control, by the way, are called oncogenes. Not that you really need to know, but onco is Greek for a hook. And onco is used for cancer because cancer is seen as this thing that gets its hooks in you. So anything that damages DNA is potentially a cause of cancer. And that's why you would want to test your lipstick using the Ames test on bacteria. If it causes mutations in the bacterial DNA, it can cause mutations in yours. This is also why there are some types of cancer that can run in families. You might have heard of a gene called BRCA1, and if you inherit the wrong alleles of that, you have an increased risk of breast cancer. That's kind of like inheriting a car where the brakes are already halfway worn out. You increase your chance of a catastrophic failure. Okay, hopefully this is all making sense. Sound off if you need me to stop or if you've got questions. Okay. Not all mutations lead to cancer. Many of them just change the primary structure of a protein. Some of them don't even do that. Because the genetic code is redundant, that means that most amino acids are coded by several codons. So some mutations will change a codon to another codon, but it's codes for the same amino acid. So it will have no effect on it at all. And some mutations will change an amino acid, but it's not in an important part of the protein. I talked earlier about proteins being like tools. Imagine you have a screwdriver. If you change the shape of the tip of the screwdriver, it has an effect on what the screwdriver does. If you change the color of the handle, it has no effect at all. Or if you change the length of the handle, it doesn't have an effect, or certainly not much of one. So some mutations don't really do anything. In fact, probably the vast majority don't. You're all carrying something on the order of 5 mutations compared to your parents. You've got an average of maybe 5 to 10 parts of your DNA that are different from what your mom or what your father gave you. And most of the time it does nothing and you wouldn't even realize that that happened. So most mutations are just neutral. Some are just about always harmful. Others may be beneficial, but it always depends on context. You can't really say whether a mutation is helpful or not unless you look at the environment that the critter is in. So I'm going to look at a couple of examples real quick. This guy, Dr. Richard Lenski, ever since 2001, he's been growing a bacterium called Escherichia coli, or just E. coli for short. And this is a really good model organism for looking at mutations because E. coli divides 6 times a day. And they have observed change over 70,000 generations. So it's easy to see what populations do over really long periods of time. I'm going to pass because I think somebody's trying to join. One moment. Anybody joining? Do we need to, the question is, do we need to know about Dr. Richard Lenski? The answer is he's a cool dude, but no. Right. Yeah. I'm not going to ask you to cough up his name on the exam if that's what you're worried about. Okay, so back to where we were. So he's found with a system like this, you can watch how genes change over tens of thousands of generations. And his estimate is that in the setup that he's got, roughly 1 in 12 mutations are actually beneficial to the bacteria. Most of them don't really do anything. Some of them are harmful, and about 8% or so actually give them increased efficiency in using their food. And he's documented a set of mutations that actually gave the bacteria the ability to use a new food source. His bacteria were growing on a sugar called glucose, and he was able to track some mutations and find out when they happened that gave the bacteria the ability to use a different food source called citrate. Even cooler, Japanese scientists discovered bacteria living in the wastewater from a nylon factory. They discovered this in 1965, and it's just called K172, or the genus is Flavobacterium. If these bacteria were wearing big clocks around their necks, they would be Flavobacterium. That was a joke. I'm assuming you're all laughing silently. The cool thing about Flavobacterium K172 is that they could digest nylon byproducts. And the reason that's cool is nylon didn't exist before 1935. It's an artificial fiber. And here you have this bacterium that's breaking down a chemical substance that, as far as anyone can tell, did not exist on the planet before 1935. The enzymes that K172 uses actually arose by mutations, and we know when they happened. It's even been possible to duplicate this in the lab. They've tried growing other types of bacteria in the presence of nylon products, and mutations have given these bacteria the ability to break down nylon. So the point is that some mutations are beneficial. They don't necessarily give you mutant X-Men powers, although I guess the ability to eat nylon is pretty amazing. But let's look at some human examples. So I'm going to jump from bacteria straight to humans, and I'll get to some that talk about human sports performance because several people here play sports or appreciate them. Since at least one music major is here, I'll actually start with a musician. That's Niccolò Paganini, such a great violinist that people thought he had signed a deal with the devil. See, heavy metal did not invent that. And he may have had, we don't truly know, but he may have had a condition called Marfan syndrome. This causes the body to produce a mutated form of a protein called fibrillin. It's dominant to the normal allele, so only one copy is needed to have the syndrome. And about 15 to 30% of all cases are caused by spontaneous mutations in the fibrillin gene. You can be born with this and not have a family history of it if that gene just happens to get a mutation in the wrong place. This guy, the front man of a rock band out of Atlanta, has Marfan syndrome and is open about it. So I'm not violating privacy, I don't think, by showing you this. Fibrillin is part of the extracellular matrix. Hope you remember that from about week 2. And it's prominent in connective tissue. So people with Marfan's have strongly affected connective tissue, especially bones. People with Marfan's tend to be very tall. They tend to be very lean, tend to have unusually long limbs for their size. In a normally proportioned person, your wingspan, your fingertip to fingertip distance when you've got your arms stretched out fully is about the same as your height. In Marfan's people, it's longer than that. They tend to have long fingers, long toes, very mobile joints. And this is livable. You can live a full, healthy life with Marfan syndrome. And people with Marfan's would be naturals at sports that require height. They'd be naturally good basketball and volleyball players, except for one thing. And I heard this thing go beep, so give me a second to... Okay, welcome to Jackson Torregrossa, who has just joined us. Okay, for Mr. Torregrossa, who tuning in just now, we've been talking about mutations and particularly about the gene for a protein called fibrillin. If it mutates, you get a genetic condition. It follows basic Mendelian rules. It's an autosomal dominant. It's called Marfan syndrome. It tends to affect the skeleton, and people with Marfan's would be naturally awesome basketball players or volleyball players, except the other place where fibrillin is really important is the walls of major blood vessels. In particular, the wall of the main artery carrying blood away from the heart, called the aorta. Your aorta is maybe an inch or 2.5 centimeters in diameter, about the size of a medium-sized egg roll. And a ruptured aorta is fatal very, very quickly. It can happen with no warning. You can feel fine. And if it ruptures, you will probably be dead before the ambulance can get there. People with Marfan's need frequent checkups. They'll go in for, heart scans and things like that. And if they catch an aorta in time, it can be surgically repaired. So with care, people with Marfan's can live full and healthy lives, except that if you have Marfan's, you don't want to strain your heart because that's puts strain on your aorta and increases the chance of a blowout, an aneurysm. So people with Marfan's are usually encouraged to take up golf or walking or something like that. But basketball is usually pretty much out. Back in 1976, basketball player for University of Maryland, Chris Patton, was playing a 2-on-2 pickup game during the summer. He went up for a jump shot and was dead when he hit the ground. Right at that moment, his aorta had ruptured, and he had Marfan's, but it was never diagnosed. Probably the most famous athlete to die of this was Flo Hyman, one of the best volleyball players we've ever had. She was on the U.S. Olympic team back in 1984, and our team won the silver. I can't remember the name of the movie, but she actually had a minor career in film after that, playing this barbarian warrior mercenary in some kind of post-apocalyptic film. And she could certainly pull that off because she stood 6'5". She was playing a game in Japan in 1986, rotated out of the game, sat down, never got up again. There was another case in 2007 of a woman on Florida State's women's basketball team who went to bed after a heavy workout and died in her sleep. Both women had Marfan syndrome. And if I had brains, I would have looked up the guy's name, but not too long ago, there was a college standout who found out he had Marfan's just before the NBA draft, and he actually got drafted honorarily, but he didn't play in the NBA, except a couple of years later, he got a medical clearance and went off to play in Europe. So several people have had this that conceivably you've heard about. Okay, so that's a mutation that affects sports performance in a way that might be harmful. It might make you advantaged as a basketball player, but disadvantaged in terms of living. But are there cases of human mutations that are beneficial? Well, here's a guy from Finland, Eero Montiranta, at the Winter Olympics back in 1964. He was in cross-country skiing, and we don't do an awful lot of this in Arkansas, but it's a hell of an endurance sport because you're working arms and legs at the same time over rough terrain. And Montiranta just crushed the competition in 1964, won several races by, I think, like 5-minute margins or something like that. He was incredibly good. And there's his pedigree chart. He is number 17, and you can see an arrow pointing that out. The fact that his square is shaded means that he had a particular trait. He had a mutation in a gene called EPOR. EPOR stands for erythropoietin receptor. Erythropoietin is a hormone that causes your body to make red blood cells. And that mutation in EPOR made him make extra red blood cells. The trait that some athletes have tried to gain by blood doping. Some of you might have heard of this. They will have blood cells transfused into them just before a game, and blood cells carry oxygen. So if you have extra red blood cells, you can transport more oxygen and you have improved endurance. And now that's called blood doping, and it's illegal. Montiranta's blood basically did that all the time. And some people that have this condition, making more red blood cells than usual, it's called polycythemia. And sometimes that goes with added risk of stroke or clotting. But his family seemingly didn't suffer any ill effects. Montiranta and his kin all lived normal and healthy lives of, usual lifespan, except that members of the family that had this trait could transport 50% more oxygen than a typical person's blood. In this family portrait, you can actually tell who's got it. Montiranta's from Finland, and people from Finland tend to be pretty pale. The kid over on the right, you can see is very pale. But you might be able to see that Montiranta has kind of a red face, and so do some other members of his family. Those extra red blood cells actually cause these people to have a more red complexion than their kin. Other than that, it doesn't do anything except it gives you incredible endurance. Mind you, that's combined with training and things like that. One gene is not going to make you an Olympic athlete. One gene does not make the difference between an athlete and a couch potato. But given the fact that Montiranta spent an awful lot of time skiing anyway and trained a lot, that allele seems to have given him an edge. All right, everybody happy? I will assume so. I'm not hearing any beeps. And there we go. One more sports gene. It's called EPAS1. You can see from the diagram in blue and green, it's got those coiled areas. Those are our old friends, the alpha helices. And then the ribbon-looking parts in the middle, they're colored red, orange, and dark blue, and they're kind of in the center, are beta sheets. So that's a diagram of what the whole protein looks like. We don't really need to worry about what it does, and I'm not sure anyone fully understands this, but it tends to make the body use oxygen more efficiently. And there's one allele of EPAS1 that in most populations is very rare, but it happens to be found in 87% of the population of Tibet. Tibet, up on the plateau back of the Himalayan mountains, it's a very high altitude country. Tibetans live at altitudes over 12,000 feet. I don't know how many of you have ever gone up on mountains that high. I have. You definitely gasp for breath as soon as you're up there. Just walking a short distance will leave you panting like you've run a mile if you're not used to it. Most human populations don't need that EPAS1 allele, but Tibetans began moving onto that high Tibetan plateau roughly 3,000 odd years ago. And people who had the allele, in the beginning, they might have been very rare, but if they had that mutant allele, they were just a bit more likely to survive and reproduce because they would have been just a bit better at using oxygen in an environment where there isn't an awful lot of oxygen. So the people who have that mutant allele leave more babies than the people that don't. Wash, rinse, and repeat for 3,000 years, and we have a nice case of natural selection in action. Same kind of deal that was going on with that. I assume you did this lab with the the maroon and the white beans, simulating natural selection on the rabbit population. I think you guys would have done that last week. This is basically the same thing going on. Natural selection makes some alleles more common and other alleles more rare. And where new alleles ultimately come from is mutation. Oh, and bringing it back to sports, there are some variants of EPAS1 that give you greater efficiency in oxygen use. And it turns out that they're more common in elite athletes that rely on generating lots of power very fast. Sprinters and weightlifters, basically, have to generate huge amounts of muscle energy in a very short period of time. And some variants of EPAS1 are more common in those athletes than they are in the population at large. Excuse me. If you have those alleles, you're just a little bit more naturally gifted at sprinting or at weightlifting. There's no one gene that will make you into a champion sprinter or weightlifter or I don't know, I guess, running back or something like that. No one gene can do that. But if you have certain variants of EPAS1, you're just a little bit more naturally gifted at it and just a tiny bit more likely to be able to join the ranks of elite athletes, people who would compete in international competitions like the Olympics. And there's one more gene that's associated with athletic performance. There are 4 similar proteins called alpha actinins, and they're involved in muscle function. I don't fully understand what they do, and we don't really need to know. There are something like 50 different proteins that are involved in muscle function, and this is one of them. This is a computer model of alpha-actinin 3. It actually consists of 2 different peptides. They're labeled CH1 and CH2. The one at the top is blue, and the one at the bottom is red. So this is showing you quaternary structure with 2 different peptides. That's a picture of a muscle fiber where ACTN3 has been stained. Those red stripes that you might be seeing are areas of a muscle fiber where alpha-actinin 3 is involved. It's in a part of the muscle fiber called the Z-plates. That black spot, by the way, is where the nucleus of the muscle cell is. That doesn't have many proteins in it, so it doesn't stain at all. The other proteins have been stained green and red. Okay. And in humans, there are several alleles for alpha-actinin 3. The normal one is usually called R, but there's a mutant allele that popped up in humans about 50,000 years ago called R577X, although people usually just call it the X allele for short. And about 50% of people of European descent carry this allele. About 18% are homozygous for it. It's a bit more common than that in Asians and a bit less so in Africans, but it does exist in every human population that's been studied. There's very few genes that are present in all members of what we think of as a race. There's very few things that really distinguish human races from each other on a genetic basis. We're all really, the vast majority of us is all the same. So alpha-actinin is found everywhere. It's found in all human populations. Some people can be homozygous for the mutant allele. Some can be homozygous for the normal allele, and some can be heterozygous. Same deal we've been talking about. That mutation that I mentioned that appeared about roughly 50,000 years ago knocks the gene out. If you have that allele, you actually don't produce any functional ACTN3. The reason is the mutation changed a codon that codes for an amino acid into a stop codon. This means that when cells transcribe that gene, but when they try to translate it, the protein stops abruptly. And you get this very short little protein that doesn't do anything. So the gene basically stops. Okay, see what I mean? Right. This would work better in person. Now, mutations in many of your muscle proteins can affect your function and can cause diseases like muscular dystrophy. My niece has a mutation in one of her muscle protein genes, and she's got a condition called CMT that is, she's going to be in a wheelchair pretty soon. Most mutations of most muscle proteins will do something like that, but this particular mutation actually doesn't have any effect or almost no effect, because if you don't make ACTN3, there's a different protein that you make called ACTN2 that can step up and take over. So without testing, you probably don't even know whether you have functional or non-functional ACTN3. However, in 2003, they did a study of Olympic class sprinters, and they were all at least heterozygous for normal ACTN3. None of them were homozygous for the X allele, for the deleted ACTN3 gene. They did a similar study in 2008 of weightlifters, and they found the same pattern. For most people, this makes no difference at all. But if you are trying to be an elite athlete, then the normal ACTN3 gene gives you a slight advantage in generating lots of muscle power fast, which is most useful if you're a sprinter or a weightlifter. I heard a beep. Let me see what's going on. Okay, not seeing anything in the chat. Everybody okay? Everybody all right? Can you see me? Okay, somebody unmute your mic just to see how we're doing. Are we there? We're good?
We're good.
All right. Okay, back to our regularly scheduled program. It turns out that mutant allele, R577X, or just X for short, turns out to be more common than you would expect in endurance athletes. That same study that found a high incidence of the normal allele in elite speed power athletes found that the X allele was more common than you would expect in Olympic class endurance athletes, long distance runners and long distance swimmers. That mutant allele is not quite as good at generating speed fast or generating power rapidly, but it does use muscles just a little bit more efficiently, and it gives you a slight edge in endurance. They confirmed this back in 2012. There was a study of 633 elite European male athletes. And by elite, I mean the kinds of people that would compete in national or international competition. The elite endurance athletes were much more likely to be homozygous for that X allele, the one that gives you a bit more efficiency. And you were even more likely to be homozygous for that X allele if you were a world-class athlete, the kind of person that could really contend for an Olympic medal, as opposed to a national class athlete that might compete within one country, but not really step up to international competition. So the better an endurance athlete you are, the likelier you are to have that mutant allele. And it gives you just a slight edge in muscle efficiency. And when this was announced, a number of companies started marketing genetic tests. They cost about $200 at the time, that would tell you whether you or your child had the regular alpha-actinin 3 or the mutant allele. And this was marketed to parents as a way of finding out whether their kid was more likely to be a champion sprinter or a champion marathoner. So, you could test your 6-week-old baby and decide whether your 6-week-old baby ought to shoot for a college scholarship in, short track events or in long distance cross country events. And evidently, there are parents that do try to plan their children's lives out that far. It's probably not money that's actually well spent, because it's only one of many proteins that are important for muscle function. And you need good function in your other systems as well. There are genes that affect not just the muscles, but the circulatory system and the nervous system that also play roles in athletic performance. Last I looked, there were at least 200 genes that have been identified that were related to sports ability. I'd be very surprised if it's only that many. I'm sure there are more than 200. And training is also important, and training and genes interact in very complicated ways. So as I've said before, there's no single gene that will make you a champion sprinter. ACTN3 is one of many proteins that are important, and all of those proteins work together. So athletic ability is ridiculously epistatic. You remember epistasis, where you have multiple genes affecting one trait? Well, you've got multiple genes affecting how fast you can run the 100 meters. And they're also influenced by, how much training you've had in running 100 meters. ACTN3 by itself does not make the difference between an Olympic athlete and a couch potato, or even an Olympic athlete and a recreational athlete in good shape. By itself, it doesn't turn anybody into Usain Bolt. It might, however, make the tiny difference between the sprinter that wins the gold medal and the sprinter that comes in fourth. If you've watched short distance track and field events, you know that the margin of victory is, sometimes just a hundredth of a second. For most people, it doesn't matter. But when you're testing elite athletes to do the absolute best that they can, that's when your ACTN3 allele might make a difference of a few hundredths of a second that separate the gold medalist from the guy who comes in fourth and doesn't get a medal at all. So for everyday life, it probably doesn't matter what alleles we've got. It does make a difference when you're right on the leading edge of sports performance, when you're somebody that can compete at an international level. But even then, it's not that important for most sports. I came across a study of judo athletes, and the study showed that there's no link between ACTN3 alleles and elite rank in judo. And if you know judo, it's not really either a power sport or an endurance sport. You're not generating huge amounts of power. You're trying to use your opponent's power against your opponent. And it's not really an endurance sport because you're not grappling for long periods of time. Judo is just kind of its own thing. And it doesn't really matter in judo what ACTN3 alleles you have. And since Casper's here, I'll also point out that I found a 2013 study that showed that there's no link between your ACTN3 alleles and elite rank in soccer, basketball, or hockey. I went looking this morning for more recent studies, and I couldn't find any my sense is that sports like that call for such a mixture of abilities that there's no single allele that always gives you an edge. And for playing 90 minutes of soccer, you need both good endurance and good speed. And you're not testing for either abilities only the way you are in sprinting, powerlifting, and marathon running. Success in those requires a mixture of different abilities and skills. So as far as I know, ACTN3 does not give you any edge in most of the sports that people play. Holy cow, that would appear to be that. I think I got through that. All right, any questions? Okay. If you've got questions, you can type something in the chat if you want to. We're actually finishing up a little bit early. I've got 10:16. These tend to go faster when I do things this way because I'm not stopping and making eye contact and interacting the way that I normally can. But if there are no questions, I'm going to go ahead and stop recording. 3, 2, 1. Okay, now I'm going to stop recording.